Okay. So, starting with the cells of the immune system. So, the precursor of all these cells that are produced in the immune system is the hematopoietic stem cell. Okay. So, first of all these immune cells. Okay. So, how does our immune system function? It is the cells that produce the molecular mediators. Okay, so these cells actually produce the molecular mediators that act upon the final target, that is the pathogen that is causing the disease, or any of the cancer cell, or any apoptotic cell. So these are the final targets, and it produces certain molecular mediators that act upon these particular targets. Okay, so, how these cells are actually produced inside the body? It is the hematopoietic stem cells that is the first cell that produces all of these. And this hematopoietic stem cell is present in bone marrow. Okay, so all these cells that are produced from a typical cell that is a stem cell or the hematopoietic stem cells. Okay, so it is a continuously dividing, that is the self-renewing cell that produces on like that on signal, on a particular signal it divides into two kinds of progenitor cells, that is a myeloid progenitor cell and a lymphoid progenitor cell. Okay, so lymphoid progenitor basically means that all of the cells that are produced as part of its lineage will be majorly taken care by the lymphoid organs. Okay, that's why the name lymphoid progenitor. And in case of the myeloid progenitor, these will directly act with the help of majorly tissues. Okay, just 1 minute. So these myeloid progenitor cells are basically helps directly inside the tissues. Okay, so the cells produced from this particular progenitor directly act upon different kinds of pathogens inside the tissue. Okay, so talking about the myeloid progenitor, it gives rise to various kind of cells, like the first is the granulocyte monocyte progenitor, eosinophil progenitor, basophil progenitor, megakaryocyte, and erythroid progenitor. Okay, so these progenitors give rise to neutrophils, okay, eosinophils, basophils, platelets, and erythrocytes, respectively. Okay, so we'll talk about these cells in detail, and this granulocyte monocyte progenitor will also give rise to monocyte that will further differentiate into macrophages. Although these cells, okay, so now we divide the immune cells into two kinds of cells. First is the agranulocytes and the other is the granulocytes. So agranulocytes are the ones that do not have any granules in them. Okay, so how do we define granules here? Granules are the structures that do not form or do not stain under any kind of staining. Okay, so for example, if we stain with any acidic or basic dyes, so agranulocytes will not be stained in that case. Okay, so those are called as agranulocytes because they are not stained and they do not have any granules in their cytoplasm. Then we have the granulocytes, that those cells that can be stained by these acidic or basic dyes and will have certain granular structures in the cytoplasm. Okay, so the example of granulocyte are neutrophils that are stained by the neutral dyes, base eosinophils that are stained by the acidic dyes, and basophils that are stained by the basic dyes. Okay, there are different functions of all these cells that we'll discuss in detail. And agranulocyte examples are macrophages or monocytes. Then we have T cells and the B cells. These are the lymphocytes. Okay. Then the megakaryocyte will give rise to platelets. So platelets are not directly involved in the immune system. These are basically involved in a clotting procedure inside the body. Right. Then we have the erythrocytes. So erythrocytes are also not involved in the immune system. These are basically oxygen carrying cells. Okay. Now going to the lymphoid cells. So lymphoid progenitor give rise to two kinds of cell. First is the natural killer cells and the other are the lymphocytes. Okay, that have the T cell progenitor or the B cell progenitor. So these T cell progenitor give rise to two main kind of lymphocytes, that is T helper and T cytotoxic cells. So these T cells are basically matured in thymus. That's why their name T. And then we have the B cell progenitor. B cell progenitor give rise to B cell. These mature in bone marrow in case of humans. Okay, just a second. So these were the lymphocytes that arose from the lymphoid progenitors, and the macrophages, neutrophil, basophil, eosinophil, platelets, and erythrocytes that originate from the myeloid progenitor. Okay, so now moving on to how do we isolate these cells? Okay, so there are two methods basically. First method is the density gradient centrifugation, that in that we separate using a histopaque 1077. Okay, so this is the name of the resin that we use for separating these particular cells, that contains polysucrose and sodium ditrizoate. So this is a reason. First, we actually form this particular column that have this histopack 1077. Okay, then we load the blood that we have collected from the patient. So first we have to load this column, and then we have to like let it rest for some time, and then we add blood. Followed by this, we actually centrifuge, and in this after this centrifugation we get some layers. Okay, so first layer is of the plasma. So this is pale yellow in color. Okay, and this will have thrombocytes, cytokines, different hormones, and electrolytes, other than the basic cells that are present in the blood. Okay, then we have a small pellet of cells that is called as a buffy coat, that will have peripheral blood mononuclear cells. Okay, so this is the buffy coat after the plasma layer. So this buffy coat will have lymphocytes and the monocytes, right? Then we have fecal histopack, that will actually have the density that is the density of this histopack. So this is the reason only. Okay, and then at the bottom we have the RBCs that have density greater than the density of the histopathy. Okay, so here we have erythrocytes and all those cells that have diameter or density greater than the density of the histopathy. So eosinophils and the neutrophils also come into this region. Okay, so the basic funda of this particular gradient centrifugation is, if we actually centrifuge using a particular resin that have a particular density, here it is 1.077 gram per ml. So all the resins, all the cells that have a density above this will be accumulated above, sorry, below this will be accumulated above, and above this will be accumulated down. Okay, so the cells, eosinophils, neutrophils, and erythrocytes have density greater than that of histopathy, so they are settled at the bottom. And the plasma and the PBMCs have a density smaller than this histopathy, so they are collected above this. Okay, so this is the basic procedure how we collect this PBMCs from human blood. Okay, so any doubts in this particular question, in this particular slide? Okay, so let's move forward. This is the question for you guys. What is the significance of density gradient centrifugation method in immunological research? So, what can be the correct answer? Please take your guesses. Okay, so yes, the correct answer is option 2, to isolate blood mononuclear cells, providing a reliable source of immune cells for experimentation. So, just like we discussed in the last slide itself, so this helps in isolating the PBMCs. Okay, that's why density gradient centrifugation is very important in immunological research. Okay, let's move to the second question. During the isolation of PBMCs from the whole blood, which of the following statements are incorrect? So, what can be the correct answer? Okay, any more answers? Okay, so here the correct answer would be option 3 only. Okay, so this is wrong. Option 3 would be the correct answer. Let's see the statement. Histopec 1077 contains polygalactose and sodium ditriazole adjusted to a density of 10.777 gram per ml. Okay, so just now we saw Histopac 1077 have polysucrose and sodium ditrozoate adjusted to a density of 1.077 gram per ml. Okay, so that's why the first statement is false. Then the second statement, leukapheresis is a procedure in which leukocytes are selectively removed from the subject's circulation. Yes, this is correct statement. In leukapheresis, what happens? The leukocytes, excess leukocytes are selectively removed from the patient body, and the rest of the components, for example, plasma, platelets, are then directly added to the subject. Okay, then the third point is Buffy coat contains monocytes, erythrocytes, and platelets. No, this statement is also false. Buffy coat contains only monocytes out of these and the lymphocytes. Okay, erythrocytes are present just below the histopech, and platelets are collected in the plasma section. Okay, so this statement is also false. So the incorrect statements are option A and C. So option 3 is correct here. Okay, so any doubts in this particular question? Okay, so let's move on to the next section. Now we'll start with the different cells of the immune system. So now we'll start with the myeloid progenitors, that are first neutrophils. Okay, so neutrophils are the most abundant WBCs. Okay, so talking about WBCs first, the cells in the bloods are basically divided into three types: WBCs, then we have the RBCs, and the platelets. Okay, WBCs, sorry, so W WBCs are basically involved in fighting the infection. Okay, the RBCs or the erythrocytes are involved in oxygen carrying, and platelets are involved in the clotting procedure. Okay, so now in this immune system part, we'll mostly talk about different cells that are involved in the WBC component. So the most abundant WBC is the neutrophils. These are predominant cells in the pus. So pus is basically the dead cells. Okay, so whenever a pathogen or any foreign substance comes, so there is a fight between two sides. First is our immune system, and the second is the pathogen. Okay, so immune system first activates the neutrophils to fight with the pathogen. So when these fight with the pathogen, there are deaths of the cell, both pathogen as well as our immune system cells. So this dead cell debris is called as pus. Okay, so the major component of this pus is neutrophils that account for its whitish appearance. Okay, then these actually are the first cells that respond to a injury. Okay, and the acute inflammation also takes place with the help of neutrophils. Okay, talking about its structure, it has a granulated cytoplasm. So as you can see in this image, these pink structures in the cytoplasm, these are called as the granules because these are stained by neutral dyes. Okay, so neutral in the sense these granules can be stained by both acidic as well as basic dyes. Okay, acidic dyes as in eosin, that is a negatively charged, and basic dyes as in methylene blue, that are positively charged. Okay, so that's why these are called as neutrophils because they can be stained by both the granules are basically antibiotic in nature that can defend the body from the infection or the pathogens. Okay, and they contain defensin, cephalosidins, cathelicidins, bacterial permeability proteins, PPI, hydrolases, and lysozyme. So these are the different components of the granules because these are bactericidal, so they secrete certain proteins and enzymes that can directly act on the pathogen. Okay, so these proteins basically create pores in the bacterial and the fungal cells that kills the pathogen. Okay, so that's why these are important at the first site of defense. Then these are the major WBCs that account for nearly 50 to 70%. So the total WBC count, out of which 50 to 70% account for neutrophils. So these are the major cells that are present in the body. These have a multi-lobed nucleus, as you can see here. This is a multi-lobed nucleus, and these are highly phagocytic in nature. Okay, so phagocytosis means that it can engulf any foreign particle. Okay, it can eat any foreign particle, and it has a short lifespan of about 2 to 3 days. Okay, it produces the active oxygen species. These actually help in inflammation. Okay, so these reactive oxygen species have a role in inflammation. Okay, then the leukocytosis and diapedesis. So whenever there is any infection inside any tissue, the circulating neutrophils will go and directly bind to the pathogen in the tissue. Okay, this we'll discuss in the next slide also. Then we have the netosis, that is neutrophil extracellular trap. This was induced by ROS, microbe size, microbial virulence factor, and the cytokines, and it composed of chromatin and neutrophil granular proteins and provide a high local concentration of antimicrobial protein or peptide. So whenever it acts upon the various pathogens, it releases a kind of mesh kind of structure that have the different antimicrobial protein or peptides as well as certain granular proteins and the chromatin of the neutrophils. Okay, and it is produced by the by inducing the ROS, microbial size, its virulence factor, and the cytokines that are produced in response to that pathogen. Okay, so this is a basic diagram of the diapedesis, how it happens. So these are the circulating neutrophils that are there in the blood vessel. It actually have some signal from the circulating immune cells in the tissue that there is a pathogen that has been entered into a tissue. Okay, so this helps this neutrophil to actually the have a process of capture, rolling, and adhesion to the endothelial cell of the blood vessel, then enter to the tissue through a process called as diapedesis. Okay, because these do not have a defined space, the so they can change their shape and they can easily ooze out from the endothelial cell and enter into the tissue. Okay, here it can actually phagocytose the bacterial cell. It can present the antigen to different adaptive immunity immune cells. Okay, it secretes the ROS and the granules and as well as the cytokines so that it can target different like other immune cells as well as it increases the inflammation. Okay, and the other thing, it can actually increase the netosis. Okay, so these are the main functions how a neutrophil act with the pathogen in the tissue. These are the different cytokines that help in its activation of the neutrophils as well as its diapedesis. Okay, so this is the process of diapedesis and how it takes place in case of the neutrophils. Okay, so any doubts in this particular slide? Okay, so let's move on to its other thing. Whenever there is defect in the reactive oxygen species production, that results in lymphadenitis. Okay, and it causes a genetic disease that is chronic granulomatous disease, that is CGD. Okay, and it is termed as a primary immunodeficiency disease. So because the ROS production is hampered, okay, so it leads to a disease called as chronic granulomatous disease. And due to this, there is enlargement in the lymph node due to infection or inflammation of the lymph node. And these patients are always pediatric patients. That means they are small and they are highly susceptible to frequent and sometimes life threatening bacterial and fungal infections, and they can be recurrent. Okay, recurrent meaning that they can be again and again infected by the same pathogen because the ROS production has been hampered. Okay, ROS production mainly takes place with the help of an enzyme that is called as NADPH oxidase, that is present in the neutrophils and macrophages also. Okay, so all the inflammation related things takes place with the help of the ROS. Okay, if we have to see what is the genetic predisposition of this NADPH oxidase mutation, then it is actually located on the autosomal chromosome. Okay, so whenever a child will have both recessive alleles of this particular gene that encodes this enzyme will have a disease, that is chronic granulomatous disease. Okay, and the dominant one would be healthy. Okay, so this is a major disease that is due to defect in the NADPH oxidase that can be present in either neutrophils or macrophages that result in lymphaditis. Okay, so let's move on to its questions. What is the primary role of neutrophils in acute inflammation? So, what can be the correct answer? Okay, so yes, the correct answer is option number two. It is the first cell that acts swiftly and respond to infections and clear pathogens effectively. Okay, so let's move on to the next question. Which enzyme is responsible for the production of reactive oxygen species in neutrophils? So what can be the correct answer? Yes, this is the correct answer. And there is a test that can actually detect how, whether a particular cell has a defective NADPH oxidase, and that test is, sorry, there is some issue with the system. Just 1 minute. So the NADPH deoxidase deficiency can be checked using a test that utilizes nitroblue tetrazolium. Okay, so this test is a gold standard test called as NBT test also. Okay, in this what we do, we isolate the blood cells and then we actually test it with a dye that is nitro blue tetrazolium and see that under a microscope on a slide. Okay, we create a smear test with this dye and then we actually see if the NADPH oxidase would be there, it would change the color of this nitro blue tetrazolium to a blue color. Okay, so that means NADPH oxidase is present there. Okay, and if the color is not changed, it leads to a yellow colored yellow color of the cell. That means that it has a complete deficiency of NADPH oxidase. Okay, and the last one, if we get any mixed population, that means that might be a carrier of the particular disease or it leads to severity of the disease. Okay, so this thing because if NADPH oxidase would be there, it would actually convert NADP to NADPH, and if NADPH would be present, then it would directly act upon this nitro-blue tetrazolium and convert this dye to blue color. Okay, so this is a test that is similar to another test that is MTT. Okay, so this is also a tetrazolium test. This actually helps in the to check the cells whether they are live or not. Okay, so this is how the test is done. Okay, so any doubts in this particular slide? So NBT is nitroblue tetrazolium test and MTT is that is based upon the methyl tetrazolium assay. Okay, so this is methyl tetrazolium test. So the dye that we use here is basically dimethyl thiazole phenyl tetrazolium. So that is a pink color dye. Okay, so this is for MTT. Okay, so let's move on to the other granulocyte that is eosinophil. Okay, so eosinophil are WBCs that are 2 to 5% or less of the total WBC fraction in the blood. Okay, it has a bilobed nucleus and it is actually stained with a acidic dye that is eosin and that is a negatively charged dye. So the granules that we have will be positive in nature. Okay, its primary function is that it is phagocytic. That means it can engulf all the uh, that is it can engulf the parasites. Okay, it has a major role during allergic condition and asthma. Its important role is during the parasitic infections. It increases upon the parasitic infection and this condition is called as eosinophilia. Okay, so it plays a major role in the parasitic infection and it increases its number upon the parasitic infection. Okay, the cytoplasm is crystalloid in nature and it contains majorly the basic proteins. that is MPPs. And MVPs consist of a toxin for helminth. So helminth are the worms. Okay, so these contain toxin for the helminths. These induce histamine from the mast cells. So after they release this MVPs for the particular helminths, they actually release different chemicals or the cytokines so that the different immune cells will get activated. So they induce histamine from mast cells. Some of the proteins can activate neutrophils and platelets also. Okay, they can cause bronchospasm. That means sudden constriction of smooth muscles in the wall of the bronchioles. Then they have two kinds of proteins or the toxin, that is eosinophil cationic protein and the eosinophil derived neurotoxin. So these two helps in declining the population of the helminth. So they directly act upon the population of the worms using these two major proteins. Okay, so first is the eosinophil cationic protein, and the other one is eosinophil derived the neurotoxin that directly acts upon the worms and kills it. Okay, so let's move on to the next cell, that is basophils and the mast cells. Okay, so basophils are non-phagocytic granulocytes and they are the least common. Even in some of the people, it is 0%. Okay, these cells help in type 1 hypersensitivity reaction. So they have a major role along with mast cells for the type 1 hypersensitivity reactions. And when activated, these release histamines and other inflammatory chemicals to the particular uh, like to in the blood. Okay, they are rare in circulation. They have large granules that can be stained with basic dyes. Okay, that have a basophilic that are filled with the basophilic proteins. Okay, that can be stained with basic dyes. That's why they are called as basophils. Okay, example of basic dye is methylene blue. Then, upon binding with IgE with Fc epsilon R1 receptor, these cells release histamine. Okay, so basophils have a receptor that is Fc epsilon R1 that binds to IgE, that is a major mediator antibody for the allergic reactions. After binding, it releases histamine. So histamine helps in inflammation. Okay, histamine basically increase blood vessel permeability and it requires smooth muscle activity. So there is a drug called as cetirizine that is anti-allergic basically. So it blocks H1 receptor and prevents acute respiratory distress syndrome. Okay, so what happens is whenever there is any allergic reaction, some people start sneezing continuously. Okay, or some people start coughing continuously. That can be due to any number of reasons, either dust, pollen, or any of the microorganisms. Okay, so our body basically basophils and the mast cells release histamine. Okay, that leads to this allergic reaction, and the drug that that we can take to decrease the histamine release is citrazine. So it decreases the or blocks the H1 receptor and it prevents the acute respiratory distress symptoms. Okay, now moving on to the other cell that is involved in the allergic reaction is mast cells. So these mast cells are present in wide variety of tissues such as mucosal epithelium, digestive tract, respiratory and urogenital tract, skin and the connective tissues. Okay, so these cells help in the These cells help in the tissues to protect them from pathogens or the microbes. Okay, they produce histamine, prostaglandin, leukotrienes that increase the permeability of the blood vessels. Why do we need to increase the permeability of blood vessels? So that whenever there is any pathogen or any foreign substance in the tissue, so all the blood cells that are flowing in the blood, the immune cells that are flowing in the blood, they can directly enter the tissue if the permeability of the blood vessels are increased. Okay, they have important role in allergies. So now we'll be moving on to the type 1 hypersensitivity or the allergic reaction that takes place in the body. Okay, so what is actually a type 1 hypersensitivity? It is a IgE mediated hypersensitivity. So the major antibody that plays role here is IgE. It basically include systemic manifestations like anaphylaxis and like localized anaphylaxis like hay fever, asthma, hives, food allergies, and eczema. Okay, and these are actually increased by when antigen induces cross-linking of IgE bound to the mast cell and basophils with release of the vasoactive mediators. Okay, so we'll discuss in detail how this reaction actually takes place. Okay, so type 1 hypersensitivity is basically happens when the receptor of the basophil and the mast cell is attached with the IgE antibody that is attached to an allergen. This reaction or the binding tend to release the granules of the basophils and the mast cells, releasing the different chemicals that are stored in these granules. Okay, so this leads to the different release of the different molecules like histamine or leukotrienes that increases inflammation. Okay, basically anaphylaxis. So this will actually lead to reddening of the eyes, sneezing, fever, slight increase in the temperature of the body. So these are the following symptoms of this particular allergy. Okay, so how does this happen? First, the IgE antibody binds to the allergen. Okay, this allergen bound Ig antibody now bind to the mast cells. So mast cell and the basophil have a receptor that is called as Fc epsilon R1. This is the receptor with which Ig antibody binds to the Fc region of the Fc region of the Ig antibody. Okay, then what happens is okay, so this antibody cross linkage of the IB. So what happens is this antibody. So how we can block it? We can block it by the binding of the antibody to the particular receptors. Okay, then there can be a chemical cross linkage of IgE cross linking the chemical. Okay, then any of the receptor mediated anti-receptor antibody can block this particular receptor, and any of then after binding there is influx of the calcium. Okay, so this influx of calcium leads to the degranulation of the different granules. So if there is anything that we can block the calcium influx, that will help in reducing this particular allergy. Okay, this granulation leads to activation of different mo- uh, like molecules inside the tissue. For example, it activates smooth muscle cell, small blood vessels, the mucous glands to release the mucus, blood platelets to actually constrict, sensory nerve endings, and eosinophils. Okay, so these release vasoactive amines. Okay, so these are the primary mediators. Okay, so the primary mediators of allergies are histamine or heparin. These increases the vascular permeability and as well as smooth muscle contraction. Serotonin, this has the same function. Then we have eosinophil chemotactic factor and neutrophil chemotactic factor that actually activates eosinophil and the neutrophils. Then we have certain proteases that help in degradation of the blood vessel basement membrane, generation of complement split products, and bronchial mucus secretion. Okay, so it increases the mucus secretion from the bronchus. There are some secondary mediators that helps in the increase of the allergy, that is platelet activating factor, leukotrienes, prostaglandins, bradykinin, cytokines such as IL-1, TNF alpha, or IL-2, 3, 4, 5, and 6, TGF beta, and GM-CSF. Okay, so these are the secondary mediators that are released further by the primary mediators to increase or amplify the allergy process. Okay, so there are certain drugs that can prevent this type 1 hypersensitivity. We have antihistamines that can block the H1 and H2 receptors on target cells. Then we have chromalin sodium. So this is a ionophore that blocks calcium influx into the mast cell so that it stops the degranulation. Then we have theophylline that prolongs high CMP levels in the mast cell. So CMP is one of the secondary messengers that actually stops the signaling. Okay, so it activates the signaling so that it degranulates, but this uh, theophylline can release or can stop this particular CMP levels so that it inhibits phosphodiesterase, which can cleave CMP to 5' AMP. Okay, then we have epinephrine or the adrenaline that stimulates CMP production so that it can bind to the beta adrenergic receptors on the mast cells. Then we have cortisone that reduces histamine levels by blocking conversion of histidine to histamine, and stimulate mast cell production of the CMP. Okay, so these are the drugs that can actually help lower the allergy or the type 1 hypersensitivity. Okay, so any doubts still here? Okay. So what happens is whenever this this is an IgE antibody. Okay, it recognizes that this is the allergen. Okay, so this binding of this allergen to IgE will now can activate the basophil or the mast cell. Okay, so these basophil and mast cell have receptors on its surface. These receptors are called as Fc epsilon R1. Okay, so these Fc epsilon R1 binds to the Fc region of the IgE antibody that is bound to the allergen. So after this binding, it activates the signaling that decreases CAMP levels and increases the calcium influx into the cells. This calcium influx actually helps in degranulating these granules that are present in their cytoplasm, releasing these vasoactive molecules like histamine, leukotrienes, and different mediators that actually acts upon different tissues to show the symptoms. Okay, so this is the basic mechanism of how a allergy takes place. Okay, is this fine? Okay, so let's move off forward. So the question is, which receptor is primarily involved in the activation of mast cell and basophils during allergic reaction? So what can be the correct answer? Yes, the correct answer is option number 4. Okay, now moving forward, then we'll move on to the next cell, that is the dendritic cell of the immune system. Okay, so dendritic cell can be aroused from both the myeloid and the lymphoid progenitor. Okay, so myeloid progenitor give rise to the dendritic cells that are present in the tissue, and lymphoid progenitor cells give rise to dendritic cells that are present in lymphoid organs that play a major role in maturation of the lymphocytes. Okay, so these the basic term meaning of dendritic cell is dendron. So dendron means branching of the tree. Okay, and these are also known as patrolling cars because these are most phagocytic cells that can engulf the foreign or the pathogenic organisms. Okay, these were discovered by Steeman, Sir Steeman, in 1970s, and it was actually he was actually provided the Nobel Prize posthumously in 2011 for their discovery. They originate from hematopoietic stem cells from both myeloid and lymphoid lineage, as I discussed, present in lymphoid and various immune organs, especially in the epidermal layer. Okay, so in the epidermal layer, we have certain phagocytic cells called as Langerhans cells. Okay, these Langerhans cells were also are derived from the dendritic cells itself. Okay, they are also present in the gastrointestinal and respiratory tract also because these tissues are very prone to infection by the pathogens. Okay, because the gastrointestinal tract is in direct touch with the food that is also coming from the outside. The respiratory tract is in direct contact with the air that we inhale. Okay, so this is also coming from the outside. So both of these are a good source of various pathogens. Okay, that's why they are also rich in the GI tract and the respiratory tract. Okay, so this is a basic structure of the dendritic cell under a electron microscope. As you can see here, it's there are various branches of this ends. Okay, so these branches give its name to the dendritic cell, that is a dendron. Okay, they have less number of hydrolytic enzymes as compared to the macrophages. Okay, these are called as a patrolling cell. That means they majorly help in signaling, so that if there is any pathogenic organism or any infection, so that it can actually activate different immune cells. Okay, so but they are phagocytic in nature, but less phagocytic as compared to the macrophages. They have few digestive bodies or the lysosome. They have poor viability, that means not so phagocytic compared to the macrophages in vitro and in vivo conditions. GM-CSF is used for in vitro generation of dendritic cells for various experiments. Okay, so what happens is we cannot directly culture these dendritic cells into the in vitro conditions. Okay, so we require certain cytokines that help the stem cells or the progenitor cells to give rise to dendritic cells. So what we do is in vitro conditions we grow the progenitor cells. Okay, we infect or we test these cells by a cytokine, that is GM-CSF. Okay, so that they can differentiate into the dendritic cells under in vitro conditions. Okay, then they can take up the antigen, that is phagocytosis, and they can induce innate immune response if they are present in the tissues, and in the adaptive immune system they can present the antigen to the T cells. Okay, so because T cells depend upon these particular cells for their antigen stimulation, they can present these antigen to the T cells through major histocompatibility complexes. Okay, so this is the major role of the dendritic cells. There are two kinds of dendritic cells. First is the plasmacytoid dendritic cells or the PDCs. They have major role under the viral infection, and they induce type 1 interferons. So type 1 interferons, IFNs, are majorly involved in the viral infections. Okay, so this PDCs are activated by the antiviral state, and it increases it activates NK cell activation for the innate immune response, and it helps in antigen presentation, and it activates n number of T cells for the adaptive immune response. Okay, moving on to the follicular dendritic cells, FDCs. These are specialized APCs. So APC is a antigen presenting cell. Okay, so these are present in lymphoid follicles. So they are important in presenting the antigen to the different T cells during their maturation. T cell and B cell for their maturation. Okay, so they bind and retain antigens by linking to complement and immune complexes, and they present the antigens to germinal center of the B cell for the secondary immune response. Okay, and they rescue bound B cells from apoptosis and induce differentiation of B cells for long-term memory B cell production or the plasma cell production. Okay, so these FDCs have a major role during maturation of B cell. That means they can create an immune response by producing the plasma cells that secrete the antibodies, and they can produce the memory B cells for the secondary immune response. Okay, so any doubts till now? Okay, So let's move further. Dendritic cells are primarily derived from which of the progenitor cells? So what can be the correct answer? So yes, the correct answer is 3. It is derived from both lymphoid and myeloid lineage. Okay, so let's move further to the next cell lineage, that is macrophages. Okay, so macrophages are the most important cells in the immune system. These are present as monocytes in the blood. When monocytes enter the tissue, or after maturation or differentiation, it turns macrophages. Okay, so these are the major phagocytic cells that helps in phagocytosis of the different antigens and the pathogens. Okay, so it helps in diapedesis, that passage of blood cells through the unruptured wall of blood vessel into the surrounding tissue, like uh, neutrophils. Okay, the activation of macrophages takes place by pathogen or by the cytokines. The monocytes and macrophages are originated from the same lineage, that is myeloid lineage. Monocytes are small, less complex, and less phagocytic floating phagocytic cells, and macrophages are large, highly complex, and these are more phagocytic in nature, and these are present in different tissues as well. Okay, the major function is they keep the system clean. Cell debris, blood cells or blood clots, and dead cells all are cleaned by the macrophages. Okay, all the apoptotic cells, different dead cells inside the body, the different blood cells that are going that have been recycled are cleared off by the macrophages. Talking about its structure, it also has certain extensions. Okay, these extensions are called as pseudopodia. Okay, so pseudopodia basically helps these macrophages to capture different pathogens like bacteria and different like foreign molecules, so that they can engulf, phagocytose, and they can be easily destroyed, and those antigens can be actually presented to different immune cells so that they can be matured and activated. Okay, these macrophages, when enter different tissues, are named differently. Okay, so in intestine, it is called as intestinal macrophages. Lungs, it is called as alveolar macrophages. In connective tissues, it is called as histiocytes. In liver, it is called as Kupffer cells. Kidney, mesenglial cells. Neuronal tissue, it is called as microglial cells. In bone, it is called as osteoclast. Okay, then like for the dendritic cells, these are also cannot be grown directly into the culture, so they are induced from different progenitor cells using a cytokine, that is MCSF. Okay, so macrophage colony stimulating factor, for various experiments in vitro. Okay, so this was the major description about the macrophages. So, which of the following statements about macrophages are true? So, what can be the correct answer? Okay, 1, 2, and 5. Okay, so the correct answer is 2, 4, 5, and 7. Okay, so let's see. So macrophages are involved in tissue repair? No, they are not involved in tissue repair. They directly target the pathogen. So they directly target the pathogen. Okay, so they are not involved in the tissue repair. They are derived from the myeloid lineage. Yes. Macrophages cannot phagocytose pathogen or cellular debris. No, they are the major phagocytic cells that are present in the human body. They can be activated by LPS, that is lipopolysaccharide, that is present as in the cell wall of the bacteria. Okay, so yes, it can be activated by the LPS. Then macrophages play a major role in maintaining homeostasis by scavenging dead and damaged cell. Yes, this is also correct. Okay, these are primarily reside in blood and do not differentiate in tissues. No, this is false because there are different tissue associated macrophages also. Okay, these produce reactive oxygen and nitrogen species to kill pathogens. Yes, this is correct. So it along with neutrophils produce the ROS. Okay, then macrophages are involved in B cell development. No. Okay, they can be involved in B cell maturation and activation, but not development. Okay, so the correct answer is option 2, 4, 5, and 7. Okay, is it clear? Okay, so let's move forward. Now we are moving on to the lymphoid progenitor cells. So till now we discussed about the myeloid progenitor cells that are involved in the immune system. Now we are moving on to the lymphoid progenitor cells. First is the natural killer cells or the natural killer T cells. Okay, so the natural killer cells are 5 to 10% in the of the blood lymphocytes. Okay, these are also granular in nature. Granules contain perforins and proteases or the granzymes. Okay, so these perforins create pores in the particular, so they create pores in the pathogens. Okay, and these proteases or granzymes basically helps in degrading the protein part of the cell membrane or the cell wall of the pathogens. Okay, they induce pore formation in target cells and trigger apoptosis. So, like perforin create pores and proteases trigger apoptosis. Okay, these have cytotoxic properties against tumor and virally infected cells through ADCC. Okay, so they directly target the tumor and the virally infected cells through ADCC, that is antibody dependent cell mediated cytotoxicity. Okay, so ADCC helps in clearing off the tumor cells and the cells or the disease cells infected by the virus. Okay, so ADCC is mediated through one receptor, that is Fc gamma R3 or CD16. So this is the name of the receptor that is present on the NK cell, so that the antibodies that can bind to the tumor or the infected virus infected cells can directly bind to the Fc gamma R3 or CD16. Okay, these NK cells senses abnormal cells and they target the cells that have reduction in MHC2, that is missing self, and unusual profile of antigen MHC1 that is virally infected or tumor cells. Okay, so what happens is any of the cells that is either tumor or virus infected will have low MHC1 count. So it decreases. Okay, so MHC1 will be as a marker of any disease. So they check this MHC1 levels on the cell surface. Okay, and it directly targets those cells that have reduced amount of MHC1. Okay, and any unusual profile of the MHC1 cells. So now these cells directly target those and will release perforin and granzyme and will kill this particular cell. So this is how an NK cell acts. Okay, so it is activated by IFN gamma. IFN gamma is a major helps in major virus infection immunity. Okay, IL-2, IL-12, and IL-18. So these are the interleukins that help in NK cell activation. Then we can test how NK cell works in a chromium release assay. So we use a radio labeled chromium and we introduce the sodium chromite into the medium and that is converted into sodium dichromate. So this is sodium chromate and this is sodium dichromate. Okay, so this helps in checking the functioning of the natural killer cells. Okay. So, how ADCC is affected? It is the density and the stability of the antigen of the target cell, the antibody affinity, and the FCR binding affinity to the antibody FC region. Okay, so this is how an NK cell function. So, based on these properties, NK cells basically are part of both innate and adaptive immunity. Okay, now moving on to a different subset of NK cell, that is NK T cells. Okay, so these are subset of the T lymphocytes. They share function of both NK and the T cells. Okay, then they have similar surface markers of both NK and the T cells. These cells express T cell receptor that can interact with the glycolipid antigen derived from the mammalian bacterial protozoan presented by the MHC1 related molecule known as CD1D on APCs. Okay, so they have the CD1D microglobulins, they have a T cell receptor kind of structure. So this is a TCR that can recognize CD1D microglobulin, a CD1D receptor from the APCs that can actually that present the antigen to the T cells also. Okay, so any of the glycolipid antigen from the mammalian bacterial protozoan receptors can be presented to the CD1D receptor to TCR. Okay, then it get activated during infection and during inflammatory conditions. Upon activation, they produce very immune modulatory cytokines and influence other cell types. Okay, for example, it leads to IFN gamma or IL-4. Okay, then they play an important role against infectious agents, autoimmune diseases, and tumors and allergens. Okay, so there are certain kind of cells that share properties with the NK as well as the T cells. Okay, so these cells are majorly involved when there is very rigorous immune response to be generated. Okay, and there is more research going on in these particular area so that how we can understand why these kind of cells are important in the immune system. Okay, so let's move forward. Which of the following statements about NK cells are true? So what can be the correct answer? So 1, 3, and 5. Okay, let's take. So the correct answer are 1, 3, 5, and 7. Okay, so let's see. NK cell induce apoptosis in infected or cancerous cell using perforin and granzymes. Yes, this is correct. So the major way how a particular NK cell helps in apoptosis is perforin and granzymes. Okay, so this is correct. The primarily respond to peptide antigens similar to conventional T cell. No. Okay, they can respond to the glycolipid antigens also, as well as the antigens from different APCs. So this is false. The activation and inhibition balance of the receptors is crucial for targeting abnormal cells. Yes, this is correct. So they actually check for the receptors on the abnormal cells. So this is correct. NKT cells do not play a role in bridging innate and adaptive immunity. Yes, this is false. Okay. NK cells can be activated therapeutically for cancer treatment. Yes, this is correct because these cells directly target the tumor. Okay. So there is NK cell therapy that in that either we can chemically induce these NK cells or we can directly provide the NK cells into the body derived from the same individual or the same patient. Okay. They do not utilize cytokines in their immune response. No, this is false because they release cytokines for their immune response. Measuring lactate dehydrogenase release can assess NK cell activity. Yes, this is correct because they require lactate for their particular functioning. So they produce lactate and they help in activity. So we can test the lactate dehydrogenases release from their cell to assess its activity. Okay, they exclusively act without any assistance from the other immune cells. No, this is false. Okay, they require assistance from other immune cell as well for the immune response. Okay, so the correct options are 1, 3, 5, and 7. So any doubts in this particular question? Not only NK cells, okay, the other cells that are involved in the organ transplant rejection are the T cells. Okay, so these NK cells will result in checking the MHC1. Okay, and these T cells will also check for the MHC1 and the 2 on the particular organ that has been donated. Okay, so not only NK will be involved in organ transplant rejection, but other cells are also involved. Okay, so let's move forward. Now we are moving on to the lymphocytes that are the other lymphoid that are also derived from the lymphoid progenitor cells. Okay, so first we have the B cells or the B lymphocytes. These cells are produced prenatally from the liver, fetal liver, and later after birth it is generated from the bone marrow. That's why the term B. Okay, so B term came from birds basically because where it was initially discovered, that is Bursa of Fabricus. Okay, it is central to the adaptive immunity and the helps in production of antigen specific antibodies, that is immunoglobulins. Okay, this was discovered by Max Cooper, and that he demonstrated that production of antibody was completely abrogated in chickens after surgical removal of bursa of fabricus. Okay, after subsequent irradiation, like I said, the B stands for bursa fabricus, not of the bone marrow, basically, because it was discovered likewise. And they are okay. So B cells are phagocytic in nature. First of all, these are termed as the antigen presenting cells. They present the antigens to a subset of T cell that is called as Th cells or the T helper cells that have a specific marker that is CD4 on them, and they are not very much responsible or essential for the cytotoxic T cells. Okay, so this is a major function, the second major function of the B cell. The first major function is production of the antibody, that is through differentiated B cells or the plasma cells. Okay, now we have a second subset that is the T cells. So these T cells differentiate in thymus. They mature and differentiate in thymus. That's why T lymphocytes. Okay, so these T lymphocytes are basically different. They are very varying in nature. So we have Th22. Okay, so to divide them, we have the major division as TC and TH. Okay, TC is C type cytotoxic T cell. Okay, so they have a different marker that is CD8, and the TH is T helper cell that have a marker of CD4. So they are differentiating markers of these two T cells. There are major sub T type subtypes, that is, so these are the subtypes of TH cells. Okay, so the TH cell, if the one is TH22, that helps in mucosal immune response, inflammatory disease, and barrier defense. Then we have TH17 that induce inflammation, promote autoimmune diseases. TH1 intracellular pathogen elimination, and TH2 that induce strong antibody response and allergic reactions. Okay, then we have T regulatory cells that maintain self tolerance. Okay, so they help in regulation of the self tolerance. Then we have TH9, it helps in B cell interaction and interaction with the mast cells and other CD4 subtypes. Then we have TFH, that antigen specific B cell immunity. Okay, so it helps in the B cell immune response amplification. So these are the different subtypes of the TH cells and how they are activated under different conditions. Okay, now what is this CD? Okay, so this CD terms for the cluster of differentiation. So these are different certain different surface proteins that are produced by different cells. Okay, these surface proteins are the cell surface receptor, signaling, or adhesion molecules. Okay, so there are different CD molecules that are present. There is the same cell express different surface proteins during development process or during activation. Okay, then overall, so these surface proteins called as cell surface markers. So cluster of differentiation termed as cell surface markers. Okay, so how they are defined as cluster of differentiation? Because a set of monoclonal antibodies that specifically bind to specific cell surface protein are known as cluster of differentiation. Okay, the cell surface protein to which set of antibodies bind are called as CD molecules or markers or antigens. Okay, that's how different cell types are different defined, because they have different set of CDs. Okay, that have a biological significance and can be used for their isolation and characterization of particular cell for types or even therapy or for diagnostic purposes. Okay, so along with it is represented along with number, and there are approximately 400 in number as of now. Okay, and as the research goes on, it can be increased to increased more than 400 also. Okay, so moving on, which marker is associated with cytotoxic T cells? So what can be the correct answer? Yes, the correct answer is option 3, that is CD8. So CD8 is basically associated with the cytotoxic T cells. Okay, now moving on to the next question. Which of the following statements are correct based on the cells that we have studied today? So what can be the correct answer? Okay, so let's see the options first. Macrophages mature into monocytes and migrate from blood to tissues. This is false. The other way around is correct. Monocytes mature into macrophages and then they go into the tissues from the blood. Okay. Then we have B lymphocytes come under the category of granulocytes category. No. Okay. B lymphocytes come under the A granulocyte category. Megakaryocytes are produced from the blood platelets and released into the bloodstream. No, the other way around is correct. Blood platelets are produced from megakaryocytes and then they are released into the bloodstreams. Okay. Then neutrophils and basophils are predominantly involved in the phagocytosis of pathogens. No, basophils are not involved in the phagocytosis. Neutrophils are involved. Okay. Leukocytosis is associated with a decrease in the neutrophils upon infection. No, this is false. Leukocytosis means production of the lymphocytes. Okay, so how can it lead to the decrease in the neutrophil? Okay, chronic granulomatous disease is a genetic disorder. Yes, it leads to it is because of the deficiency of NADPH oxidase. So it is a genetic disease. Dendritic cell link innate and adaptive immunity. Yes, this is correct because Dendritic cells are present in the tissue itself, that is, that have the first line of defense, and they also help in maturation and increasing the memory and the secondary immune response. So it is present also in the adaptive immunity. Then macrophages have poor viability than dendritic cells in vitro. Yes, this is also correct. Neutrophils are in circulation and keep circulating. No, this is false because they have a lifespan that is of 2 to 3 days. Okay, so the correct options are F, G, and H. That is option number 2. Okay, so any doubts in this particular question? Okay, if not, we'll move forward. What role does okay, H point. Macrophages have poor viability than dendritic cells. Okay, so what happens is there are two conditions here. First is in vitro and during in vivo conditions. Okay, so the dendritic cells have a lifespan of more than, I think, 10 to 15 days. Okay, so they remain in either the tissue or in the lymphoid organs based upon their function, right? The macrophages do not like move into the blood. They generally move into the blood and they move to different uh, like they move to in vitro conditions. That means the dendritic cells grow very wide in vitro as compared to the macrophages. Okay, so let's move on to the next question. What role does genetic similarity play in syngenic mouse models? So what can be the correct answer? Okay, 2 and 3. Okay, so the correct answer is option 3. So syngenic mouse model means that have genetic similarity. Okay, if there is genetic similarity, then if there is any successful cell transplantation, then it would not induce any immune response, and that would be without any immune rejection, aiding research on immune cell function. Okay, so if there is genetic similarity, then it would actually decrease the risk of immune rejection if there is any cell or tissue transplant. Okay, so that's why third option is correct. Okay, and for the second option, it enhances their immune response, providing a model for robust immunity. This will be false because if we transplant any of these tissue from the other mice to this particular mice, then it should not evoke any immune response. Okay, that's why syngenic mouse models are used. Okay, so let's move on to the next question. What is the primary advantage of creating a single knockout mice before double knockouts? So what can be the correct answer? Okay, so if there are any single knockout mice, so that means there is modification into a single gene. Okay, after creating this modification, if we create a double knockout, that means knockout in two genes, so that will be called as a double knockouts. Okay, so rarely we study two genes interaction like that. So the primary advantage of creating a single knockout mice is the breeding strategy is simplified. Okay, we create a hetero mice and breed those to get a homo colony of the single knockout mice. Okay, so like we create a, this one will be a normal and this will be a knockout. Same case with the mother, this will be a knockout and this will be normal allele. After we breed those, there will be one progeny that will have a both the knockouts. Okay, so it is easier to create a single knockout mice. If we have to create a double knockout, then we have to check whether, uh, like, based on the crossing over pattern and the probability of getting both the knockouts together in a single mice, that would be difficult and rare phenomenon. So that's why it simplifies the breeding strategy for achieving the double knockouts, making genetic studies more efficient. Okay, so the correct answer is option number 2. Is the explanation clear? Okay. So this was the last question for today. If you guys have any doubt, you can ask. Okay. If you guys have not, if you do not have any doubts, then we'll close the session for today. Thank you guys for joining in. We'll meet next week. Thank you.